That was a wonderful talk and an excellent prelude to our next speaker, Dr. Tim Greten, who is a tumor immunologist as well as a gastrointestinal oncologist and a senior investigator at the National Cancer Institute. So of course we're very happy that the government is not shut down so that he can be here today to present his laboratory, translational, and clinical research experience on immunotherapy for biliary and hepatocellular cancers.
Okay, thank you very much for the very nice introduction. Thank you for giving me this title. It's a true challenge. If you go and look what's known about the immunotherapy and biliary cancer, I could just say nothing and we can go back and we're done. It's the second last talk, you're all exhausted anyhow. I haven't even really published any data that I wanted to share with you. I was forced because I think some people knew that we're working on this, so I'm going to share actually some public data that I didn't want to share with you. And on top of that, I'm standing here, and I have to please patients, so they understand this. The basic scientists that want to actually understand something new, and then on top of it, the clinicians who want to do the clinical trials. So it's going to be a difficult balancing act in the next 20 minutes, but I'm trying my best. So one thing I have to mention that the NCI. So as you heard, I work for the government, so I never get any money, but the NCI has a CRADA with AstraZeneca that basically supported one of the studies that I will report. And so for those of you who don't know the NIH, it's basically a huge research institution. It's the largest research hospital. So and we basically do have a regular hospital. The only difference is we don't have an emergency room because all the patients that come to us are basically patients that come by invitation and are coming for clinical trials. And if anybody's interested, I'm not going to go into any of the details now. We don't want to bore everybody, but happy to discuss this later. But what we really do, and there's a lot of people working in the program here dedicated to liver cancer and cholangiocarcinoma. And the important thing I want to mention for all the patients is everything I talk is experimental therapies. So I'm going to give you some background in the immunotherapy because obviously there is a huge hype in immunotherapy for liver cancer. I will update you on some of the results that we and others have obtained. Mainly, it started really in HCC and then it kind of shifted over to cholangio. But most importantly, I will give you some perspectives into the future and show you what we can actually learn, I think both from clinical studies and then basically going back into the lab and what you can expect to come in the future. So it all started with this paper that came out in 2010, which was really a breakthrough when anti-CTLA-4, so one of the first checkpoint inhibitors developed by Jim Allison, was tested in melanoma patients, and all of a sudden you can see that there was a slight difference, and everybody's very proud of this, what everybody talks about, this long tail of the curve that patients actually all of a sudden survived a disease where there really hasn't been any progress, hadn't been any progress until 2010. The next big part was testing anti-PD-1. Again, this was in melanoma. And you can see here the data that was published then 5 years later, again in the New England Journal. To be honest, my personal breakthrough was actually the LEX study. We participated in this study, and I think this is really a milestone study because even if you doubt that immunotherapy may work, I think this will actually prove or show you that it works. And this is a study where now anti-PD-1 treatment was done in patients with MSI-high tumors. And you can see all of a sudden that we actually only need a very small number of patients. You can see the outcome, the changes in tumor size on the top. You can see that number one is the important thing is, the majority of patients actually respond. It's not only 15 or 20%, it's actually really many. And you see a survival curve. That looks pretty good. Obviously, this is not a comparison, but I can tell you a lot of these patients that were treated on this study, and I treated some of them, I knew what their outcome would have been without the treatment. And this is really, I think, the most important data that we have. And in the context of cholangio, yes, there were actually some cholangio patients, I marked them for you, included. And I think, even if this is only a small proportion of patients, the The one point everybody has to make sure is that their tumors are tested for this marker because of this clear benefit. This is FDA approved across the board for any type of cancer, and it's something that you really shouldn't miss. Now, let's go back to the biliary cancer. So there really, as I said, is very little known. So we tried to summarize this 2 or 3 years ago and what we know about biliary cancer, and there are some descriptive studies that look at immune cells, and the idea is that obviously in the context of inflammation, these tumors develop, which suggests that immunotherapy may be of relevance, and there have actually been a few studies, very, very small experimental studies, published using different approaches, vaccines, or then obviously a case of adoptive T-cell therapy and others. But to be honest, there really isn't a lot known. What we know, there is a lot of obstacles. We know that there is tumor promoting factors, as you can see, or anti-tumor effector cells that we can use, such as CD8 cells, NK cells, that we can use to kill tumor cells and have anti-tumor efficacy. But then there is also a lot of immune suppressor mechanisms that we actually find in the environment that really inhibit this, and that's why the patients progress. So let me now go and tell you very, very briefly, I'm not going to go into this because I know we don't talk about HCC, but this is how we started, about a concept that we first tested in HCC, where we tested this anti-CTLA-4, in this case anti-tremelimumab, in combination with ablation. So this is kind of the rationale for this study. So what, if you believe in immunotherapy, and I think if you see this one slide that I've shown you with the PD-1 and the MSI high tumors, where really all we do, we activate T cells, and we know now why it works, I think it's probably the best proof that we really have that immunotherapy can work. So if you believe in this kind of approach, you do need a T cell that recognizes a tumor. So basically, this is the T effector cell, and this T effector cell is specific for a tumor. It recognizes an antigen which is expressed by this tumor and can kill this. Now, there's many approaches, and we have been doing this for many years. I've been doing this for way too many years now, to induce these T cells. There are cancer vaccines that have been done, some transferred T cells, and checkpoint blockade is obviously one approach. But the approach that we used was actually using already established treatments such as tumor ablation, which interventional radiologists do all the time, to cause a wound and an infection in the or an inflammation in the tumor, and then use this in combination with checkpoint blockade. Obviously, this is a beginning. You can see, I already mentioned, there are many mechanisms why this actually may not work. All these suppressors mechanisms have not been targeted with this. But anyhow, we started this in HCC, and I'm going to go through this quickly. We tested this. You see here the outline. There are, and again, this is a proof of concept. It's a small study. There is not, we don't have, there's no way that I can prove that the ablation really did something to this. But the bottom line is, this type of approach seems to work in HCC, which is also, if I was just a lame person, I could say it's also liver cancer, so it should actually also work in cholangio. What's the difference there? And the interesting thing, and I want to come back to this, is that, and again, that it may be very obvious, it actually only worked in those patients where we find CD8 T cells or T cells after treatment in those tumors. So if you look here on the right, we looked at the patients that responded to the treatment versus the non-responders, and you can see only if we get these T cells, which obviously if you believe in the T cells to recognize them, they have to be there, only in those where we actually can find T cells in the tumor, this works. If for reasons that we're trying to understand, you can't get the T cells in the tumor, this treatment is not effective. So based on this study, we said, okay, so if this works in liver cancer, why doesn't it work in the other liver cancers? So we started this in biliary cancer, probably way too naive. We used the same approach. You can see here the trial design again, the same treatment. In this case, the ablation was a microwave ablation. And I don't want to bore you with all those details, but these are the patients that we usually have. Patients received multiple different types of treatments, including different chemotherapies, radiotherapy, etc. What I can tell you is that the treatment was relatively well tolerated. Now you may say this is a huge list of adverse events, but this is actually, these adverse events are not only related to the treatment. Obviously, a lot of the patients had already issues to begin with. And you can see here now the outcome if we look at the change in tumor size over time. And the bottom line is, yes, we do find some lesions, but it's nothing where we can say, this is a home run. It really, it works in all the patients, like I have shown you, for instance, for the MSI patients. You see that we have some patients, and in those cases, it also works actually for a very long period of time, which is, always something very, very encouraging. But at the same time, we also have to be always critical, I think, with our data. And I think it's fair to say that there is a signal, and we can say that we are probably on the right path. But that's pretty much it. Now, I want to give you, and I think this is both for the scientists as well as directed to the patients, give you some information about, so what are we doing now? It's a negative study. Okay, so now there's one, you could say, okay, we just do the next one, and this one didn't work. What I like is actually use the data from the negative studies to learn something so we can develop our next, better trials. So the other point I want to make is immunotherapy is very different than cytotoxic therapy. And this is just one example of a lesion or a patient and then the patient's lesion, where you can see it actually took a long time for the lesion that is here growing in the beginning to respond over time. And this is something that I have seen not all the time, but sometimes you see this in patients, that in contrast to the chemotherapy, the immunotherapy actually takes some time, and you see first a progression, and we can potentially at one point to discuss what type of progression this is, but sometimes these type of treatments actually take some time. And the other point is now the productive, what we can get out of this. So you've seen similar slides before now about the immunohistochemistry. Now these are actually slides from tumor biopsies that we got from these patients that were enrolled on the studies, and where we look at this. And I discussed this together with our pathologist, and he doesn't find any T cells in these tumors after treatment. So this is after treatment. And whereas I've shown you in the HCC field, it's very simplistic thinking. You find the T cells and they respond, but here we don't find them. And obviously, the stroma around it may just form a physical barrier for the T cells to go in. In contrast, and I don't want to go into the details how we do this, we also draw a lot of blood and study the T cells from patients and do next generation sequencing. And we looked at the effect of the treatment with the checkpoint inhibitor to see how the clonality of the T cells change. And that's very similar in HCC. So it's not that the drug doesn't do anything. We see indeed a change in the clonality, a decrease in the productive clonality upon treatment, what we would expect, and it's similar to treatments where it does work. But it appears that in the local environment, there must be something different, and obviously we're working on this and trying to address this. And this physical barrier is something that I just want to put out there as an hypothesis why and a treatment that may be working right now in HCC is not as effective in cholangio. So where do we go? Well, there are obviously ongoing, and this is actually a typo, it should say cholangio, it's not only HCC, because I actually changed this slide. So obviously there are immune checkpoint inhibitors, and there are multiple investigators in the audience that do these studies testing immune checkpoint inhibitors. More importantly, a lot of investigators test immune checkpoint inhibitor combination therapy studies. But then there is a lot of interest in antibody treatments, genetically engineered T cells, and vaccines. And then something that I would like to tell you a little bit more in the second part in my talk about changing the tumor microenvironment, because I think that's something that so far has been not really paid enough attention to. Before we go there, so what do you do in an environment like the NIH? So if you see that your treatment studies are not effective enough, obviously you have to go back to the mouse models. And this is one approach that we're currently testing, or that we have tested. Otherwise, I wouldn't have the data yet in combination with immune checkpoint inhibitors that we tested in the lab to test an IDO inhibitor. which is one way to potentially target regulatory T cells and dysfunctional T cells in the environment. And without, again, showing you all the details, in this case now we switch. Now we're treating four-leg individuals. And you can see the tumor growth of these liver tumors here. We do see some effect of the, in this case, anti-CTLA-4, and we have similar data with anti-PD-1. But then the combination with, in this case, an IDO inhibitor seems to be very effective, both in different models that we have tested where we grow these tumors subcutaneously and orthotopically. And this is obviously one approach that can go into the clinic. But the way more interesting part is this one, and that's the gut microbiome. So a lot of you may have heard that there is a lot of researchers that believe that you are actually only probably less than 90% of yourself, or 10% of yourself, I should say, because the majority of your genes and cells and et cetera, how you want to calculate it, are bacteria. And these bacteria, they don't make you sick. They live with you. We call them commensals. You can find them in your gut. You can find them on your skin. You can find them anywhere. And it's actually not only bacteria. It's also viruses and other individuals. So we kind of live with them together. And so just to give you just a very, very brief introduction. So I told you in our body, the microbial cells, they outnumber the human cells by a factor of 10. A lot of these can be found in the intestine, where you find, and then if you look at the genome, it becomes even more complex. And it is known, and there's multiple and different studies from scientists way better than our own research, to show that that they can affect multiple different biological mechanisms, and the development of the immune system is dependent on these. So we started studying the microbiome actually a while ago. And so what I'm going to show you now is, so what I should tell you is there's different ways to study the microbiome. And from an experimental point of view, one way to very easily study this is basically, and this only works in mice, don't try this at home, is to treat individuals with an antibiotic cocktail. So you pretty much eliminate more than 99% of the bacteria in the gut. And we tested this. And we tested this in different mouse models, and I had two very nice introductions for my talk, so I don't have to tell you any more about hydrodynamic injection and what plasmids to use for cholangio, but it's exactly what you have heard before. The only difference now is we utilize these models, and either we treat these mice with antibiotics that we keep in the drinking water or not, and the result is very obvious and clear. You can see this here. You can see on the left the macroscopic tumors, and maybe the picture is not large enough, so on the right you can see the number of tumor nodules that we can count on these livers. And you can see that in this model for intrahepatic cholangio, there are significantly fewer tumors by treating the animals with antibiotics. So we got very interested in this because obviously, this is could potentially, if we understand the mechanism, this could translate into a potential treatment strategy. I should say this is a very specific effect, and again, I'm just going to give you show you some of the data that we have. It doesn't work in the lung, so we'd use a different model with the same treatment, and in this case, you can see there is not fewer tumors, and we also tested subcutaneous tumors. So the mechanism that we see with this antibiotic treatment is limited to the liver, but obviously if you talk about and work on liver tumors and cholangio, this is obviously very relevant. So then we asked ourselves, obviously, why is this? And in my own group, we do something that we do pretty much all the time when we do our research studies, is we do basically the same that we do when we treat patients. We perform immunomonitoring. So we look at the different immune cell subsets that we can find in the periphery, as well as the ones that we can find locally in the liver. And this is the result. So we looked basically in the liver of these mice treated with the antibiotics. And you can see in red are basically the number of cells per gram tissue. And I know there's only a few immunologists, so I don't want to, I'm going to explain this to you. So we look obviously at, T cells that I have talked to you before and other cells. But the interesting thing that I want to say here is there's an accumulation of so-called NKT cells, which is a very distinct cell population that not that many people really take into consideration, because this is a very, very rare cell population if you work in mice or if you work in human, with the exception of the liver. So the liver is actually, from an immune point of view, a very different organ. I have shown you down here the relative distribution, and just ignore mainly what type of cells these are and just look at the color code, but compare peripheral blood and liver, and you can see it's very different, the distribution of different immune cells in the liver from other parts of the body. The other part is there's obviously a lot of immune cells in the liver, and it actually turns out that these NKT cells that I've just mentioned are actually pretty prevalent in the liver. Now, I have to tell you a little bit about these NKT cells, because otherwise it's hard to understand how this works. So this is a mix. They share properties of both T cells and natural killer T cells. They recognize lipids as antigens, so it's different from the conventional T cells. They recognize these lipids, which are presented on CD1D MHC molecules. I told you that they are very abundant in the liver, and they have a conserved T cell receptor, which helps us identifying the cells. And the important point to remember is that these cells actually express a certain chemokine marker, CXCR6. Now, going back to our model, we looked at CXCR6 positive cells in antibiotic-treated mice. And to make a long story short, you can basically see that most of the NKT cells are CXCR6 positive. That's number one. And number two, down here, if you look again now at CXCR6 positive cells in the liver in antibiotic-treated mice, there's a clear increase. So it appears that if we treat the mice with the antibiotics and kill all the bacteria, there's an increase of NKT cells. Now, how can we prove that the NKT cells are really relevant for the effect that we see? And then when I talk about effect, I mean the anti-tumor effect. So we used two different mouse models. And basically, these are two different mouse models that don't have NKT cells. So one is a CD1D knockout mouse, and the other one is a CXCR6 knockout mouse. And in both cases, you lose this effect of antibiotic treatment. So if you treat now these mice with antibiotics and you have tumors in the liver, it doesn't work anymore, suggesting that the NKT cells are indeed relevant effector cells for this mechanism to work, because otherwise, you don't see a difference between antibiotic treatment or not. Now, the last part, and I actually, again, have to thank the previous speakers, because liver anatomy was already a little bit introduced. I have to tell you a little bit more about the liver anatomy, which is, again, very different from other organs. So one of the important factors is obviously that we have the portal vein. Now, the portal vein is a vessel that basically brings all the dirty blood that comes from the gut with all the trash into the liver. So it's actually there's a good idea why bacteria may actually affect biological mechanisms in the liver, because it's really the first filter or the first organ the blood has to go through that drains the large and the small intestine. You already heard about the liver, how it is organized. Now, there is one important part to remember. So if the blood actually comes in this portal vein and reaches the liver, the first cell that the blood really hits here is what we call the liver sinusoid endothelial cell, or LSAC. So that's a special endothelial cells. And what we did is now we actually looked at the cell and looked for markers that could explain how the NKT cells are upregulated. So how does this work? Well, chemokine receptors have specific ligands. And I told you that the NKT cells that I talked about are CXCR6 positive, and the ligand for CXCR6 is actually CXCL16. Okay. So what we did is we looked for the CXCL16 in the liver of mice treated with antibiotics, and that is what you can see here. You basically get an upregulation of this particular chemokine in the liver sinusoid endothelial cells derived from mice treated with antibiotics, and you don't see this in the rest or in the rest of the hepatocytes. So what I propose, and this is already, what I skipped is that, we also showed this obviously by immunohistochemistry. So what I propose is the following. If you treat a mouse with a broad mix of antibiotics to kill a lot of bacteria in the intestine, that changes to mechanisms that we're trying to better understand, but what it leads to is it leads actually to an upregulation of liver, of the expression of CXCL16 in the liver, or more specifically in these endothelial cells, which are really the first barrier that the blood has to go through when it is coming from the small intestine and going into the liver. This upregulation of CXCL16 then will lead to an upregulation or an influx of the CXCR6 positive NKT cells. And these NKT cells can be used as an alternative or in addition to CD8 T cells that I've shown you when we use, for instance, checkpoint inhibitors to treat tumors. And the reason why this works very well in the liver and not anywhere else is twofold. Obviously, the chemokine expression is regulated by the bloodstream from the liver, so it's kind of the closest. That's number one. And number two, I showed you that these NKT cells are cells that are usually overlooked by a lot of people because they are very rare, but you find them to be very abundant in the liver and nowhere else. And we're currently working on these mechanisms in more detail because this is actually a very nice and elegant way. If we can modulate this in patients, this would be, I think, a very elegant way to really mount an anti-tumor response against tumors growing in the liver. And then, obviously, there are multiple ideas what you can do in terms of combining this with other treatments. So with this, I would like to close and thank my team. Chimar is the postdoc working on the bile duct studies that I showed you. Sujun Zhang is another postdoc working on the IDO study that I showed you. And then the clinical team and a former clinician working with me who now has his own group back in Ireland, who worked on the clinic, who worked with me in the clinical trials, and obviously support from many others. And with this, I would like to thank you for your attention. And I guess I forgot to mention all my collaborators, but this was mentioned before. You can't do this without teamwork. Thank you.